So let's start the last session that we have on session 4. We see here in evening here in Waterloo, which is device and technology for healthcare and communication and photonics, which covers the UN SDG goals like 3, 9, and 11. So we have a speaker, Professor Allison Tong, Principal Research Fellow and School of Public Health at the University of Sydney Nano member as well. Professor Tong.
Well, thank you. First, my name is Allison Tong. I'm actually from the Sydney School of Public Health at the University of Sydney. And I'd like to thank the organisers for the opportunity to present on some research work that we did to look at research priorities for sensor technology to address COVID-19. So my presentation is probably going to look a little bit different, a little less technical compared to the other presenters earlier today. So just by way of background, from the 19th of November, just yesterday, there's been more than 55 million confirmed cases of COVID-19 and over 1.3 million deaths across 220 countries. And I'm sure we all know that the control of COVID-19 transmission and the clinical management of patients with COVID-19 remains a major challenge globally. And advances in nanotechnology and also the Internet of Things has really stimulated the proliferation of sensor technology. And for sensor technology, it can be used to detect viruses or their compounds, antibodies, and other chemicals or physiological parameters to help inform public health interventions and also the clinical care of patients who are in the acute but also the recovery phase of the disease. And we know that recently there's been some evidence around long COVID and patients experiencing symptoms and complications in the long term. So with sensor technology, the main application in the context of COVID-19 has been used to detect febrile individuals using infrared sensing devices. However, there are some limitations with these technologies, including the high rates of false negatives or positives, and it's not so helpful in individuals without fever. It's also been mainly used to detect the presence of viral RNA using the PCR tests, but they still are expensive, resource-intensive, quite cumbersome to use, relatively slow, and provide little information about the transmissibility or the virulence of the virus. So the talk today is really going to place emphasis on co-production. So here, we embarked on an initiative to co-produce research priorities for sensor technology to be applied in COVID-19. And the vast majority of projects have really been researcher-driven, with very little or no input from patients or the broader community. And there really is a need to allocate resources in a more strategic and targeted manner to address the problems that are of greatest importance to the community. And this can help to improve the relevance and use and impact of the findings. Just looking at what's been happening in COVID-19, there's actually very few studies or initiatives that have conducted research priority setting partnerships, particularly those including patients and members of the general public. And after doing a search in sensor technology, I don't think there's any priority setting being done in looking at sensor technology. So we commenced a process where we brought together scientists, people from engineering, medicine, across diverse clinical specialties, including infectious diseases, cardiovascular disease, and so on, members of the general public, and also patients. So bringing all the kind of key groups together to co-produce research priorities. And the importance of co-production is increasingly recognized as an important part of this process. And this was an article published a few years ago in Nature, actually, where they highlighted the importance of co-production right from the beginning, from the proposal to the final publication, and I'd even extend beyond that to not only dissemination, but also translation and implementation of the findings. And just pulling out a quote here from the paper, which said, people living with conditions and those who care for them provide context and counterpoint to unchallenged scientific wisdom. So for the specific project that I will be talking about, To provide some context, this was a joint initiative of Sydney Nano, which is led by Ben Egerton, I think he presented earlier today, and also the Marie Bashir Institute, led by Tanya Sorrell. Our focus was on the development and the application of sensor technology for the prevention, detection, and management of COVID-19. The intended beneficiaries were the patients with confirmed or suspected COVID-19, health professionals, and also the general public. There were a few aims to this process. So firstly was to identify those shared research priorities, which were termed as research statements, to understand the reasons for their priorities, and also to generate some ideas for implementing them. So there was 2 phases to this process. First, we conducted a survey, and then this culminated in a workshop where we discussed in more detail the priorities. So in terms of the survey, we developed this with the steering committee that was part of this project, which included multidisciplinary team members. And we sent this survey out to the collaborators, and this did include members of the public, patients, as well as clinicians and researchers. And we asked all the participants to rate the importance of each of the research statements on a 1 to 9 scale. So any rating from 7 to 9 was indicative of that being of critical importance. So here you can see the mean scores, the median scores, and also the proportion of the participants who rated the statement to be of critical importance. So we had 43 people complete the survey. And so the top statements, the top prioritized statements were to develop a point-of-care instant screening test for COVID-19, to detect how infectious a person with a virus is, to identify the level of immunity a person has to COVID-19 or its change over time, to develop tests to assess people's immunity against the virus, to identify who needs a COVID-19 test in those who may be asymptomatic, to develop a non-invasive, quick, cheap, and effective diagnostic test that people can do for themselves, develop a detector for airborne virus, and also identify who needs a COVID-19 test in people with a chronic condition and may have symptoms similar to COVID-19. So these were the statements that had a mean of 7 or more, a median of 7 or more, and more than 65% of the participants rating it to be of critical importance. So we brought these top rated statements into a workshop where we had 65 people attend. So in this workshop, the 6 breakout groups were presented with the top prioritized statements, and each group did have a facilitator to work through a number of questions. All the sessions were recorded and transcribed, and we coded the transcript using hyper-research to identify themes, really focusing on the reasons for the priorities. And I'll just walk you through the themes that were identified. So again, just to remind you of the scope, where this is really about sensor technology to detect infection, infectivity, and also to assess immunity. So the first theme was around giving higher priority to research that would enable efficient clinical decision-making. So things that would prevent delays in patient access to treatment, not just patients with COVID-19, but patients who require ongoing medical care, to preserve resources in that clinical setting particularly, and also to provide prognostic information. And for each of these, I will show you a couple of quotations just to highlight these points. So a clinician said, the amount of time and energy that's expended on people not knowing whether they're infected or not is inordinate and disruptive and enormous. An instant point of care would be of enormous value. You want to use personal protective equipment on patients that you know is going to be the highest risk, so you can preserve resources. The next theme was around minimizing societal disruption and its consequences. So to allow a return to normal life and also to reduce stigma and isolation, particularly for patients who did test positive for COVID-19. The economy being closed down is in large part because you can't actually control what happens when you walk into a factory, when you walk into a place of work, And so a point of care, very rapid test is what would actually make it possible for people to return to something like normal functioning. Every time I mentioned that I have COVID-19, this is a patient, that I have COVID-19 to somebody, they all jumped out of the way from me, that I've had it. They all jumped 5 meters away from me. They don't want to talk to me. And that was back in March, that when I had it. And just to let you know, I mean, this workshop was run in August. And this patient was still encountering that sort of stigma and those sorts of reactions. The next one was around prioritizing research statements such that it would protect the community. So to trigger contact tracing to establish safe environments for the community, to safeguard particularly those vulnerable populations, gauging individual susceptibility, and also managing risks, particularly among healthcare workers. You don't want to wait for somebody to actually develop the clinical disease and to be diagnosed and then for the tracing to take place. That you have the opportunity to, for example, close down that supermarket or get the team in to deal with the aged care facility very quickly. People who have chronic illness, they are certainly very concerned and worried about how vulnerable they are, and so things that give them security and answers to people from some of these groups would be very welcome. And then the next one was about being prepared for the next phase of the pandemic, considering that the development of immunity and also the development of vaccines that are currently happening, and also to maintain suppression. COVID is evolving. That's really important for us as a research team and developers in terms of what we look at. The next wave, or not so much the next wave of infection, but where are we heading with this? So is it towards those who's immune and those types of activities? Where are we going to be in 18 months' time when some of our research comes out, and what's going to be the most productive at that point? And another clinician said, we've gone through this first wave on adrenaline, but we're really going to need to find out how to sustain a response in the long term. And the final one was around implementation, and this was focused more on the feasibility and acceptability of sensor technology. So I'll go through this one in a little bit more detail. So the participants or collaborators said for the compounds used, they should be minimal in risk in collecting it. So the use of urine, for example, may be preferred because it's non-aerolizing. That it's easy to obtain, recognizing that compounds such as sweat could be difficult to collect. Saliva may be most feasible for assessing immunity. They also suggested that sensor devices need to be affordable and cost-effective, be convenient, non-invasive, have high accuracy, and have long battery times because, for example, recharging devices may be impractical in very busy clinical environments. And then also there were some concerns about ensuring the protection of privacy, so them suggesting that there must be appropriate governance to protect people's privacy, and this would help to ensure uptake. In terms of digital platforms, AI and machine learning, they said it would require rapid transmission of data packets and would require international collaboration for adequate data to generate algorithms. And I thought I'd just compare this with another recent article that was published, and this was a figure that summarizes features of an ideal biosensor required to be developed for effective use in pandemics. And I think most of the points are quite similar. But I suppose in our workshop, I think there was some focus on it being non-invasive for it to be acceptable for use. Then we identified extracted ideas specifically for the compounds, devices, and also general application of sensor technology in COVID-19. I won't go through all of these in detail, but just highlight a few examples from each of the research statements. So for developing a point of care test for COVID-19, was suggested to target a different type of sample to detect other compounds or chemicals that the body generates due to infection, and this could be used as a signature of disease, detecting viral components, using microfluidic technology, developing wearable non-intrusive devices for healthcare workers that capture temperature and other clinical parameters with data captured in a central service for monitoring, using devices in the toilet to measure microbiome, and so on. The next statement, detect how infectious a person with the virus is. So there were some ideas around developing face masks with sensors so they might change colour if a person is still contagious. Using sensors to measure physiological parameters, breathing, inflammation, cough. Developing cell cultures with cell types that are very susceptible to being infected by the virus and to expose them to infected people to measure that transmissibility. Identifying the level of immunity a person has to COVID and its change over time. So developing a saliva test that might detect spike protein as capture medium for immunoglobulin. An idea around sort of a needle device that's inserted into the arm similar to the one used for the continuous glucose monitoring that could be useful for ongoing tracking and monitoring. Measuring serological responses or T-cell responses for assessing people's immunity. And in terms of identifying who needs a COVID-19 test in people who are asymptomatic, so using surveillance technology and monitoring sewage systems, which I think is happening in some regions. The final two, to develop a non-invasive, quick, cheap, or effective diagnostic test that people can do for themselves. So maybe the use of breathalyzers, using blood samples, respiratory secretions for home tests. A detector for airborne virus, so using detectors, say, in air conditioning symptoms to detect viruses in, say, high density places or high risk places, such as nursing homes. Use of animals. There's been some news articles around the use of trained dogs to detect COVID-19. And finally, identify who needs a COVID-19 test in people who have a chronic condition. So developing a device to perhaps look at variability in physiological parameters for those with chronic conditions or with conditions that have similar symptoms to COVID-19. So just to summarize, the top research priorities that came out of this process was really around the need to have a rapid point of care testing available to assess the infectivity immunity of people. And it was interesting to compare, there seems to be lower priority for physiological monitoring, particularly for those with COVID-19. And the reasons underpinning their priorities are around to inform efficient decision-making, particularly in clinical settings, to minimize societal disruption, to protect the community, particularly those who are vulnerable, and also to be prepared for the next phase of the pandemic. And what was emphasized was that the sensor technology had to be easy and feasible to implement, to be acceptable for use. And I suppose just trying to emphasize and highlight the value out of the co-production process and the involvement of patients and their family members and the general community, it did broaden the focus on immunity or immune status. So there was a lot of emphasis, particularly among patients, around stigma, isolation, and mental health. Recognizing that there's the development of vaccines and that the research should be responsive to that, and also to address some of the uncertainties of that long-term immunity and also reinfection. This work was done in Australia, so we do also recognize that further work should be done at an international level, and this co-production initiative can help to formulate a more equitable, coordinated global response to future pandemics and other threats. And finally, this process, it does provide a roadmap for allocating resources to the most critical areas of need. It does offer ideas for innovation in developing sensor technology to strengthen that whole of society response to the COVID-19 pandemic and potentially help to prepare us better for future pandemics. And I'd like to close by acknowledging the coordinating team and all the collaborators who were involved in this project. Thank you very much.